Well, good morning, everybody, and welcome to what is really a special Grand Rounds presentation. Did, Timothy, did Susan tell you afterwards we're going to have 4 chairs up here for the panelists? So as soon as the last people gets grab 4 of those.
Got you.
Today for a very special Grand Rounds presentation in honor of Constance Lieber. As you know, Connie Lieber was a heroic figure in the history of mental illness research. And in order to pay our respects and honor her memory and her legacy, given the fact that in the aftermath of her passing, there have been numerous types of memorial events, the most fitting thing to do would be to have presentations by some of the exemplars of the research which she worked so hard over the years to support, and hence today's program. I want to first welcome the Lieber family who are with us today. Her husband Steve, daughter Janice, son Sam, daughter-in-law Mary, also Jeff Borenstein, who's the president and CEO of the Brain Behavior Research Foundation, or as we refer to it, the organization previously known as NARSAD. This is an organization which everybody who is in the field of psychiatric and mental illness research has some connection to, and I think gratitude for, because it's an organization that has supported everybody in some way at some stage of their career. Over the years, going on 3 decades, Connie and Steve Lieber have been integral parts of it and have really guided and supported this organization in the most substantial and constructive and generous of ways. I also want to welcome, although he's not yet here yet, but will be soon, Dean Goldman. Dean Goldman is a cardiologist, but came to know the Liebers and I'm sure admires them as much as anybody who's in the field of psychiatry for what they've done for the field and for Columbia. And as I speak, here he is. Good timing, Lee. Connie Lieber was diminutive in size, but she was a giant in spirit, energy, courage, and intelligence. And while she may not have received the same degree of acclaim and notoriety as some other people who were advocates for medical research, I regard her as somebody who is every bit as deserving of that stature and akin to, let's say, Mary Lasker was to Congress. Connie has been to mental illness. She's worked tirelessly for our cause, and we're really forever indebted to her. I have 2 photos I want to show that I kind of exemplify. This is Connie with, does anybody know who this is? Think Beautiful Mind. This is Sylvia Nasser, Columbia faculty member in the School of Journalism who authored The Beautiful Mind about John Nash. And this is in 2014 at the American Psychiatric Association here in New York, where Connie and Steve were honored for all that they've done for the field of psychiatry. Now, our program today consists of 4 presentations by individuals who were closely connected to the Lieber family and their efforts. Eric Kandel, psychiatrist, neuroscientist, Nobel Prize winner. Jonathan Javitch, who's the Lieber Professor of Psychiatry at Columbia and the Director of Molecular Therapeutics. Alice Medalia, a cognitive neuroscientist who directs the Lieber Recovery Clinic at 51st Street. And Herbert Pardes, the former CEO of New York Presbyterian Hospital, head of the Scientific Council of the Brain Behavior Research Foundation, and the person who served as a mentor and guided the Liebers in their philanthropic endeavors over the years. Now, in order to maximize the time we have, we're not going to take questions after each presentation, but we will have a general question, answer, and discussion session at the end in which I'll invite all the presenters to come up and on the stage and hold forth for questions. There is going to be a lunch following the presentations, as we usually have, but unlike the usual, it's not open to everybody. We can't fit everybody, so it's by invitation. So you haven't received an invitation, I'm afraid you're on your own afterwards. Without further ado, it's my pleasure to introduce Dr. Eric Kandel to kick off the proceedings. Eric?
I'm delighted to participate in this memorial symposium for Connie Lieber. And it gives me an opportunity to express my own personal debt to Connie, to Steve, and to Herbert Pardes. Connie was special. Anyone who knew her loved her. And what really struck me about her was the depth of her intellect. She was interested in art, she was interested in music. She knew a lot of biology, particularly developmental biology, which fascinated her. And when she heard you discuss science, particularly when she found something that was interesting, she was just marvelously engaging. She had a long-standing interest in development of the brain and in disorders that begin very early during development. Steve and Connie were married for 65 years, and they were inseparable. And they really started psychiatric philanthropy in an important fashion with the Brain and Behavior Foundation in 1987. And for this, they sought, understandably, the advice of Herbert Pardes. Herb is an absolutely remarkable guy who has been a significant influence on me throughout all of my career. And he has been particularly important to the Liebers. He was the advisor from the very beginning, and he led them in the right direction at every single point. And I've benefited from many aspects of his involvement. He was head of NIMH, he was then head of this department, then became dean at the medical school, and then became head of the hospital. In each case, he really did a fantastic job. He's just an extraordinary academic leader, and he's been a personal friend who's helped my career in many important ways. I received 3 grants from the Liebers through the Brain and Behavior Foundation, whose founding and current president is Herbert Pardes, the person who introduced me to the Liebers in 1994. And I had the privilege of introducing Jeff Lieberman to the Liebers, took them all out to dinner, and that got the relationship between Jeff and the Liebers going. Each of the 3 grants that I received from the Liebers was critical for the evolution of one phase of my research. My first grant in 1995 allowed me to open up a new front. I'd spent my whole career up to this point on studying learning and memory in a simple invertebrate, marine snail aplysia. And with the help of this second grant, I was able to open a second front to begin to explore similar problems in the mammalian brain using gene knockout experiments. And as a result, my laboratory and that of Suzuma Tanagawa were the first ones to use gene knockouts to study mice and look at the molecular mechanisms of memory storage, and we sent it back to back at the Cold Spring Harbor Symposium. The second grant allowed me to really change the way genetic experiments could be carried out in the mouse. Prior to this, all knockouts were general. If you knocked out a particular gene, you knocked it out throughout the brain, and you had no control over it at all, when it was turned on, when it was turned off. With the help of the second grant, Mark Mayford in my laboratory was able to revolutionize the study of genetically modified animals by developing methods for regional and regulated expression. He used a CAM kinase promoter, which restricted expression to the forebrain, and if you generated a number of lines, you can get even more restricted expression, for example, as I'll show you later on, to the striatum. But in addition to using a promoter that restricted it to the forebrain, he had that promoter drive a tetracycline transactivator. That's a bacterial transactivator that doesn't obviously exist in the human brain. And so you can take the promoter and fuse it to any gene and have that gene expressed by the tetracycline system. And the interesting thing about that is that this can be turned off by deoxytetracycline. So if you turn the gene on and then inject the oxytetracycline into the animal, you can turn the gene off. So for the first time, you had regionally specific expression, and it was temporally regulated. And the third grant from them helped my colleagues Eleanor Simpson and Christoph Kellendorf to apply Mayford's methodology to develop a new and powerful animal model of the cognitive negative symptoms of schizophrenia. This is Eleanor Simpson, who I think is here, and Christoph Kellendorf, who probably is here as well. And they were in my lab at this time when they began this project. And they were struck with the fact that PET imaging at Columbia had pinpointed dopamine in the striatum in patients with schizophrenia. Now, one had known since the discovery of antipsychotics that dopamine is importantly involved in an excess way in schizophrenia, but the idea that the striatum is particularly important was relatively new, and Columbia was one of the contributors to it. And people had shown as increased dopamine synthesis, increased dopamine release, and increased D2 receptors. And so what Eleanor and Christoph did was to use the Mayford methodology and asked what are the consequences for cognitive prefrontally involved symptoms of schizophrenia of overexpression of D2 receptors in the striatum. So they used CAM kinase promoter, fused to tetracycline, and drove D2 receptor. And they expressed D2 receptor only in the striatum. When they gave the oxytetracycline, they could turn the gene off. So this was very nice. So they used a number of cognitive systems that you could use to study the cognitive, the prefrontal symptoms in schizophrenia. And they focused particularly on the T-maze, which is a very simple task, very easy for the animal to perform, has 2 steps to it. First is a force run, in which a mouse has to run a T-maze in which one arm is shut off, and it runs successfully. Of course, it gets the reward on the left side. You then open up the T so the animal can go either way, and after 4-second delay, you put it back into the T-maze, and now the animal has to remember they got the reward in the left arm previously, and now it has to go on the right side. And with the gene on, you see that it takes the animal much longer to reach criterion than it would if it was a normal wild-type animal. So clearly this was deleterious. And the interesting thing is that when you turn the gene off, you had the same defect. What does that mean? That means it's possible that it's not the adult expression that is the only thing that's important. In fact, the primary thing that's important. It's the developmental time course of this, that having excessive D2 receptor expressed through development is enough to give you these cognitive symptoms. So the 2 of them tested this directly. They repeated this experiment, limiting overexpression to the striatum only to the prenatal period. At birth, they immediately gave the oxytetracycline. And this was alone to cause this persistent cognitive deficit. So this indicates that the cognitive deficit in working memory in the prefrontal cortex results not from continuous overexpression of D2 postnatally, but from excess expression during prenatal development. This is something that fascinated Connie, because Connie was particularly interested in what happened in the brain during development. And this provided some of the early evidence that these cognitive symptoms developed during development and what happened later on. This is one of the reasons that blocking D2 receptors in the mature adult has no effect on these particular symptoms. The conclusion is changes in striatal D2 receptors alter cortical-dependent cognition. This may explain why D2 receptor blockade in adulthood does not ameliorate cognitive symptoms. This is concordant with the fact that schizophrenia is a developmental disorder, and the striatal D2 receptor expression has a greater impact on cognitive functions than was previously assumed. This may be due to the fact that D2 receptor overexpression in the striatum also affects D1 receptors in the prefrontal cortex, which are critical for working memory. When Christopher left our lab to set up his own, Eleanor had to look for new collaborators, and she examined a number of candidates, found none of them suitable, and she realized that no single person can replace Christoph, so she recruited 2 of them, Ingrid and Aurelia, and together they focused on the negative symptoms of schizophrenia. Negative symptoms, and we did this in collaboration with Peter Balsam, has three components to it: decreased motivation, social withdrawal, and blunted affect. And we focus primarily on decreased motivation. Now, let me just give you an example of what this involves and what we're trying to model here. If you take a typical schizophrenic person and you tell them, look, I'd like to invite you to our house at 116th Street and Riverside Drive. Denise will make a wonderful lunch for you. They will say, oh, 116th Street and Riverside Drive, that's such a schlep. I'm not interested in doing that. If you somehow pick them up and take them to 116th Street and up to the 5th floor, they sit down to the meal, they enjoy it as much as you and I do. So they have difficulty not with the hedonic enjoyment of it, they have difficulty with planning the route to go there. They find that very difficult. And so we thought we'd try to explore this in the mouse. So we can expose a mouse to two things: home chow, which tastes awful, and milkshake, which they love. Now, in order to get milkshake, they have to work a little bit. They have to press a bar. You know, it's a little bit like going to 116th Street. And under normal circumstances, they do it. But if you turn the transgene on, this is too much for them. This is milkshake in a wild-type animal without the transgene on. And if you turn the transgene on, they forget about the milkshake. They eat this crappy chow because they don't have to do any work in order to get it. When you turn the transgene on, they go back to enjoying the milkshake much, much more. So let me summarize the D2 motivational deficit. Less willing to work for preferred outcomes. Hedonic reactions are intact. Less able to accurately represent the value of future outcomes. How does this relate to the clinical picture? This is people. Anticipation, very weak in schizophrenia. Consumatory behavior is the same. I must tell you, when the Liebers opened their institute at Hopkins, I presented some of these data. And somebody walked up to me and said, you know, my son acts exactly like your mouse. That was the highest compliment I've ever been paid in my scientific career. What are the mechanisms whereby striatal D2 overexpression alters cognition, anticipation, motivation? So this is what Eleanor has carried out recently, beginning with a collaboration with Joachim Roper. She did with something which is very difficult. She recorded the firing patterns of the dopaminergic neurons in the midbrain. And for this, she used in vivo electrophysiology, recording single units. They confirmed the dopaminergic neurons in the ventral tegmental area in the substantia nigra. She confirmed this biochemically. And this shows you what the firing pattern is in control animals. Brrr, 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 these characteristic bursting patterns. If you increase D2 receptor expression, that changes completely. It fires less frequently and no longer fires in bursts. Thus, D2 receptor overexpression has a dramatic effect on firing pattern. And it shows that the postsynaptic receptor can control the presynaptic firing pattern of neurons whose cell body is some distance away. Do these changes in dopamine cell firing in the D2 receptor overexpression affect dopamine release? So dopamine release can now be measured in vivo in real time in behaving mice using fast scan cyclic voltammetry. And Eleanor did this. She used a carbon microelectrode implanted in the striatum and a specific pattern of voltage applied to the electrode. This results in a current that is proportional to the amount of dopamine at the electrode. This method is too invasive for humans, but it works extremely well and can be safely used in mice. So in a Pavlovian conditioning experiment, she used it to measure cue-evoked dopamine release in the D2 overexpressing mice and in controls. So she gave one conditioned stimulus followed by milkshake and another conditioned stimulus that was not followed by anything, so unreinforced. And you can see this is the reinforced and this is the unreinforced. In the wild-type mice, if you overexpress the D2 receptor, you see a significant reduction in the amount of dopamine released. And controls show different response to CS and US by session 6, while here it takes much longer. It takes session 7 to show effect. And the learning results in dopamine release in the conditioned stimulus. It takes longer for D2 overexpressing mouse to learn because early in training, there's a terrific deficit in dopamine release. So this deficit This difference here is much greater than difference here. This is responsible for the fact it takes the animal much longer to learn. So let me summarize what we've learned from this fast scan cyclic voltammetry. The D2 overexpressing mice are slightly slower at learning from positively reinforced cues. There's some evidence for this in patients with schizophrenia. The dopamine release in response to reward presentation is normal. It mirrors the normal behavioral response to reward presentation in D2 overexpressing mice and in patients. And the dopamine release in response to cues that predict reward is lower early in training, but is normal after learning, suggesting that training can compensate for this dopamine deficit. So let me summarize some of the conclusions. Overexpression of D2 receptor limited to development alters cortical function and cognitive performance in adulthood and contributes to cognitive symptoms. So this shows that just overexpression during development without having any expression later on, as you can do artificially, leads to cognitive symptoms. Concurrent overexpression of D2 receptor in the striatum also results in behavior related to negative symptoms. An increase in postsynaptic D2 receptors alters the firing pattern of midbrain dopamine neurons and alters the striatal dopamine release during learning. Conclusions about animal models in general. Mice can be used to model molecular perturbations involved even in highly complex diseases like schizophrenia. Animal models can be used to determine the developmental time windows of pathological changes. And quantitative behavioral analysis can be used to dissect highly specific clinical symptoms. Animal models allow us to explore intact circuitry using measures that are too invasive for clinical studies. Let me end with a personal note. I was very grateful and very much helped by the Liebers. So in my Nobel acceptance speech, it was, of course, only natural for me to mention the Lieber support. And I am the only American psychiatrist who's ever gotten the Nobel Prize. So I thought I was the only American who's gotten the Nobel Prize who's been supported by the Liebers. But then when I looked at the record, I saw that even non-psychiatrists were supported by the Liebers. Paul Greengard, my friend with whom I shared the Nobel Prize, was independently supported by the Liebers. So two people who won the Nobel Prize have been supported by the Liebers. That's a good beginning, and we have people sitting in the audience who are good candidates, so who knows what's going to happen. Thank you very much. Sound?
No. Well, this is thanks to all the new security we have around here. It'll probably take me a half an hour. Oh. Magic. Okay, it's a pleasure to be here to share some of our research with you. Before I start into the science, I just need to also take the opportunity to express my enormous gratitude to Connie and Steve. They've as well had an enormous impact on my research career. I haven't received the Nobel Prize, but I actually received one more NARSAD award than Eric, so it's, didn't realize that until now. I started younger, so I was allowed to get a Young Investigator Award. So I was funded by 2 Young Investigator Awards early in my career that had a huge impact on getting me started, as well as an independent and then a Distinguished Investigator Award. I've also benefited immensely from the Lieber's generous support of the Lieber Center here at Columbia, as well as it's an honor every day I get to see my signature that I have their name in my title, thanks to the endowed chair. And they also played a huge role in helping my move over from PNS to Kolb a few years ago and helping us to set up our microscopy facilities, which are benefiting both us and many of our colleagues. So the impact has been immeasurable. But Connie loves science, so I'm going to talk about science. So to get to the present, I'm going to go back into the past. Over 40 years ago, the discovery was made that there's a remarkable correlation between the potency of antipsychotic drugs for the dopamine D2 receptor correlated with their antipsychotic dose in people. And the correlation's even better if you actually look at the free concentrations of the drug in the brain. We therefore knew that blocking D2 receptors is a critical part of antipsychotic activity. This was discovered independently by Phil Seaman and Saul Snyder in 1976 and was actually a critical aspect of why I actually went to Hopkins to do my MD PhD with Saul. I was astounded, and frankly to this day, I'm still astounded that there can be this kind of quantitative correlation between a bimolecular interaction between a drug and a receptor and something so profound as the human experience of hallucination or a delusion. It's been difficult to figure out how this works. You just heard aspects of it from Eric, and I'm going to tell you a related but a different story. Our understanding of the regional differences in dopamine in the brain in schizophrenia have really been dramatically enhanced over the last 10 years, in large part thanks to the work of Anissa Abidargam, who was here until recently. And I'm going to focus on two aspects. This is a pre-commissural dorsal caudate shown down here, which is the, what we can also call the rostral caudate, as opposed to the ventral striatum down in this ventral area. While the lure had been that it was the mesolimbic, the ventral area that actually was overactive in schizophrenia, what actually turned out when Anissa's group, led by Larry Kegelis here, discovered was that it's actually the rostral striatum, shown here, which has the big increase in dopamine signaling. In contrast, it's much smaller in other regions, and in fact, in the ventral striatum, there's no significant elevation of dopamine. So this was a bit of a surprise. It allowed us to start to think about the fact that the brain's not a homogeneous receptacle for dopamine, that there are actually different circuits, different neurons that are reacting in different ways, and then we had to start to pull apart this circuitry. We know for sure that there's this profound correlation between the level of synaptic dopamine and response to antipsychotics in those patients who do respond. This is showing the correlation between high dopamine predicts a very good response to drugs. But at the same time, something very fascinating emerged in this study that I just told you about, which is that there's actually a inverse relationship between negative symptoms and dopamine levels in the ventral striatum. So the opposite, in a sense, is true in the ventral striatum. The less dopamine you have, the more inclined you are to have negative symptoms. So this really presents a huge pharmacological conundrum. We know that we need to block D2 receptors to promote antipsychotic effects, but we also know that negative symptoms are a problem in schizophrenia, and it appears that actually blocking D2 receptors in the ventral striatum may in fact worsen negative symptoms based on this understanding. We can expand this context beyond just schizophrenia. Studies have shown a correlation in ventral striatal dopamine receptors in a number of disorders involving motivation, shown here with reduced receptor binding in drug addiction, ADHD, and obesity. And there's a robust rodent literature showing that dopamine and dopamine T2 receptors are critical in the nucleus accumbens, the ventral striatum region, for incentive motivation in rodents. Jalan Solomon was been a pioneer in this, but many labs have contributed. That's primarily been done by lesion studies or by microinjection of drugs, which in fact show that D2 blockers in the ventral striatum do worsen motivation in rodents, consistent with this conundrum that I'm raising. So a number of years ago, we decided to set about this, trying to use some of the genetic types of tools that Eric was just telling you about. We used a first-generation virus to increase expression of D2 receptor. Now, there's a critical difference that I need to separate this from what you just heard about. We're doing this in the adult mouse. So a lot of what Eric told you, he highlighted, was a result of development. And development plays a critical context. Even if you can turn something on and off with the receptor, it may be related to the fact that it's on a developmental overlay. So you have to sort of do a separation between what you just heard and what I'm going to tell you about. In the adult, we're now overexpressing D2 receptors selectively in the ventral striatum. And Pierre Triflieff led this project in the lab a few years ago with a number of colleagues who are here, who I'll point out later. And what we saw in the same kinds of tests that Dr. Kandel just showed you, that these mice in which we increase now D2 receptors in the adult actually are more highly motivated and will work harder for a reward. So in the adult setting, it seems like that's consistent with what I'm showing you here for the lesion studies and for the patient studies. This is a first-generation virus, and it expressed everywhere where we injected in the striatum. So we wanted to move to a more refined approach where we put the receptor where it belongs, which I'll show you in a second. But this also introduces the problem of how complicated it is to think about pharmacotherapy when you're targeting dopamine receptors. You know, we've known about this for a long time. Why haven't we made more progress? Well, there are a lot of reasons. Dopamine receptors are on multiple types of cells in the striatum. They're on postsynaptic medium spiny neurons of two flavors, which I'll tell you about in a moment. They're on cholinergic interneurons, but they're also on the terminals of corticostriatal glutamate neurons and on the terminals of dopamine neurons. And they actually play, in some cases, opposing effects. Turning on the receptor can lead to decreased dopamine release from the dopamine neuron, but in fact, obviously, dopamine will activate postsynaptically. So how does one target with a drug? We already have the problem we'd like to target one region and not another, but the receptor is the same. And then we also have receptors that are in different localizations. So these medium spiny neurons are 90% of the neurons. They're the projection neurons from the striatum. But they come in two flavors. They come in neurons that are called the direct pathway that express D1 receptors, in the indirect pathway that express D2 receptors, and also adenosine A2A receptors. They have different circuitry. The direct goes directly. The indirect has a way station. And in general, the direct pathway is thought of the go pathway. If you turn on these neurons, you enhance locomotion and behavior. The indirect pathway is thought of as a no-go pathway. If you stimulate this pathway, the animal's locomotion will decrease. While most of these studies showing the differentiation between the direct and the indirect pathway have done in the dorsal striatum, there are analogous direct and indirect pathways in the nucleus accumbens, the ventral striatum, as shown here. There are also, interestingly, collaterals that project from the D2 indirect pathway to the D1 so that they can regulate each other here. Also down here, which is, where's my mouse? There are also collaterals down here, which Christoph Kellendunk has studied, which is a fascinating story, which I'm not going to talk about today. So what we're talking about now is overexpressing D2 receptors very selectively in just this population that normally expresses D2 receptor, but raising the levels. And we did this with a viral strategy related to what you just heard, where we can drive expression, in this case, by using a specific promoter driving a Cre recombinase, which will cause the viral expression to start expressing only where it's expressed, and by using a mouse that's expressing Cre only in D2 receptor expressing neurons or only in A2A, which is basically the same pathway, we can actually selectively express the receptor in this group of cells, and we see it. I'm not going to go through the details, but we see expression in D2 expressing neurons and not in the D1 or the cholinergic neurons. And we have a very robust increase in expression. This is about a 3-fold increase in expression. So we really, truly are overexpressing the receptor. So just as an aside for the aficionados in the group, there are a number of very powerful techniques which have emerged to manipulate circuits in recent years. There's optogenetics, which uses activating or inhibiting light-activated or inhibited channels to turn on or turn off receptors with exogenous light. It's proven a powerful tool to look at circuit analysis. There are designer receptors which only react to engineered drugs, which can be both inhibitory and stimulatory, which have also been profoundly helpful to do circuit dissection. And when you put in, actually it's an analog of clozapine that's used, clozapine N-oxide, which is inert at normal receptors, it activates or inhibits these pathways, and we can learn something. There, you're always using exogenous control, light or the ligand. In this case, all we're doing is increasing the amount of the receptor that's normally there, which increases its sensitivity, but it increases its sensitivity to endogenously release dopamine. So if we look at normal behaviors, we maintain the spatial and temporal control of dopamine signaling, but enhance the effect of the signaling. So these are all parallel methods that allow us to look at something different. So in a progressive ratio test, just like Dr. Kandel just showed you, the mouse has to press the bar to get the reward. It increases exponentially, so before you know it, it has to press 500 times or 1,000 times to get the reward. We see that overexpression of the D2 receptor now in the adult mouse in the ventral striatum lengthens the survival time. It keeps pressing longer because the test goes for either 2 hours or until the mouse doesn't press for, I think, 3 minutes. The mouse that has the overexpression of the receptor has dramatically more lever presses. It's more willing to work for its reward, but the press rate is the same. So these animals are more motivated. They're also locomote more. This is an open field, and you can see that the distance traveled by the mouse overexpressing the D2 receptor is enhanced, and that's quantitated here. That's not a surprise. Remember, I told you the indirect pathway is the no-go pathway. The D2 receptor is inhibitory. If you inhibit a no-go, you go more. So that's what we expected to see. But now that we had developed the system to see these robust behavioral outcomes in an adult mouse, we could actually start to try to understand the signaling that's responsible for that, which we think is critical to delve more deeply into developing more efficient and targeted therapeutics. The dopamine receptor, as you know, is a G protein coupled receptor. What it does is in response to dopamine, it signals to G proteins. It activates the GIO family of receptors, which inhibits adenylyl cyclase and leads to a decrease in cyclic AMP. Through the G beta gamma subunit of the heterotrimeric G protein, it also activates potassium channels if they're there. It can also inhibit calcium and sodium channels. It also has coupling to voltage sensitive channels in the terminals. These are all the G protein signaling, which have been the classical way we've thought about G protein coupled receptors. What we've learned over the past perhaps 2 decades from the work of Robert Lefkowitz and others is that there's another G protein independent signaling arm of GPCRs, and that's mediated through this protein called arrestin. It was named arrestin because it was originally discovered to arrest signaling. It binds to the receptor and blocks its ability to talk to G proteins. But it later turned out that it's actually critical for receptor internalization and that it serves as a scaffold for different kinases and phosphatases, and thus certain signaling can take place through an arrestin-dependent pathway as well. And Mark Krohn's lab has shown that it signals the D2 receptor in an arrestin-dependent way, signals through AKT and GSK3 beta. Now, functional selectivity is a term that's a big rage in pharmacology now. It's based on what we've learned is that in particular receptors, we can actually selectively activate these pathways with certain ligands. While certain ligands will activate all the downstream pathways, other ligands, through mechanisms that are under active study, can actually just activate G protein pathways or just arrestin, or in some cases, can selectively identify and target different G protein pathways. So there's a lot of work to try to identify drugs that can do this, and there's been success in opiate receptors, which is really interesting in terms of differentiating therapeutic and side effects. In D2 receptors, while there's some interesting compounds, none of them have the specificity that allow us to do this, and furthermore, we're still strapped with this problem of the receptors being on all these different places where they have opposing effects. So we decided to take a different path. Could we make receptors that actually were biased in themselves, and by selected mutations based on what we understand about the molecular interactions that lead to signaling, could we make an arrestin-biased receptor that would signal to arrestin and not to G protein, or vice versa, to G protein and not to arrestin? So Prashanta Thamseti, a pharmacology graduate student in my lab, set out to do this a few years ago, and he succeeded. So this is an arrestin-biased mutant, and you can see in this assay, which measures arrestin recruitment to the receptor, it's nearly like wild type. It efficiently recruits arrestin, but when you look at the G protein activation assay, it's completely dead. And his G protein biased one, in contrast, is just like wild type in terms of recruiting G protein, but is completely dead for arrestin. So this provided us a really powerful tool to start to move to the in vivo setting to, for the first time, differentiate what the signals are to lead the different behaviors. And there are other signaling in vitro that I'm going to skip over that shows that downstream at cyclic AMP and internalization, this breeds true, the selectivity follows. So to show you that this actually is true in the brain, here we're taking D2 knockout animals. They have no D2 receptors in the entire brain, and we're putting back the receptor only into the ventral striatum in the indirect pathway. And if you do that, whoops, I'm leaning over my touchpad. The green is the control because there's no D2 receptor and there's just a control virus. There's no activation of G protein as measured here in this radioactive GTP binding assay. And if you put back wild type receptors, you get this nice activation curve. The receptor works again, it activates G protein. But if you put in the arrestin biased receptor, no G protein activation. This is what we expect. This is an arrestin biased one. So now we can actually go do the experiment that I'm telling you about. Here's what I showed you before. The wild type receptor enhances locomotion when you overexpress it. So I'm going to take a show of hands. If we put the arrestin biased receptor in here, it doesn't do any G protein signaling. This G protein coupled receptor can't signal to G proteins. Does it enhance locomotion? Who thinks it's going to enhance locomotion? Not very many people. Jeff, the smart guy over here. It enhances locomotion. Phew, lucky thing, since that's what he voted. This is really quite amazing. It shows that a non-G protein mediated pathway is having this profound behavior. Now, this is a situation where we are overexpressing the receptor in the wild type animal. So there still is G protein signaling by the endogenous receptor. So let's take a more extreme case now. And let's go to the knockout animal, and now there's no D2 receptor, so the only D2 receptor that's there is the arrestin bias receptor. Again, you see completely wild-type receptor-like locomotion, and we can also look at cocaine. If you give cocaine, it blocks reuptake. You get a boost in dopamine. In the knockout animal, you have hardly any response. In the wild-type receptor restored here, you have a profound increase in locomotion, and it's identical in the arrestin-biased one. So this is telling us that locomotion is mediated by arrestin signaling. Now, this doesn't show it's through AKT. It's something we're trying to do, but it's really quite remarkable. Lefkowitz doesn't want to call these G protein coupled receptors anymore. He wants to call them 7TM receptors. Here's an example of G protein not mattering. But what about for the other behavior? When we look at motivation, it's the complete opposite. This arrestin biased receptor has no effect on increasing lever presses. It has no effect on breakpoint. Again, the pressing rate is unchanged even with the wild type receptor. So the overexpression enhances locomotion, but it doesn't enhance motivation. So this is really fascinating and allows us to think for a moment about a classical problem with psychostimulants, which is separating activation from motivation. Psychostimulants increase locomotion. They increase behavioral activation. Is this what's happening, or are they doing something more specific to motivation? I think this is the first time where I actually can see that an animal that in fact is behaviorally activated is not more motivated. And you can see a little hint of this if you look at the survival curves. Remember I told you if the animal presses even once in a 3-minute period, the test keeps going. You see an intermediate effect here when you look at the survival curve of the session duration. These mice are activated, and they're more inclined to once in a while go and press the bar. The test keeps going, but they're not motivated to press for the reward. They don't press more. These are just a few presses that keep them going. So we're really starting for the first time being able to separate activation and motivation. So in summary of what I've told you, biased receptors can be used to probe distinct signaling pathways in a much less ambiguous way than traditional approaches and allow us to look very specifically at their behavioral correlates. That overexpressing the arrestin bias receptor in the adult animal leads to enhanced locomotion but not to enhanced motivation. That only arrestin signaling seems to be sufficient to get the locomotion. And we're now moving on and studying the G protein bias mutant. We're trying to understand what are the downstream mediators of this arrestin signaling and trying out constantly to think about the implications of this for therapeutics and how we can tap into this system at different levels. So let me just thank the people who did this. Today's the Eleanor and Christoph show because all the work that I've talked about with the second generation virus has been a very close collaboration with Christoph Kellendunk. Eduardo Gallo, who is a postdoc in both of our labs, has done the behavioral work and also electrophysiology work that I haven't talked about today. Pierre did the first generation virus work in collaboration with a number of valued colleagues here at Columbia. Bo and Aneko made the virus, and Prashant created the biased receptors, and Ying actually is a typo, sorry. Ying actually is now carrying on with some of the biased work, trying to make an even improved G protein biased one. Collaborators helped us with the GTP gamma S, and Kim helped with the knockout animals because they had enough of those animals bred up. And thanks to our support, in particular, the Lieber Center. Thank you very much. I'll let you pull it out.
So you have been hearing how Connie promoted research in our understanding of the basic biology of psychiatric disease, but she also understood that people need to be treated in the here and now. Her goal was to eradicate psychiatric disease, but she also wanted those currently living with illness to receive the best treatment. And she campaigned for research and services that promoted productive lives for people with mental illnesses. Remembering Connie and her impact on science means to know her in the context of her family. That's how I knew her, a mother who came to me with Steve and asked if I would offer cognitive remediation to improve her child's cognitive skills. You see, Connie was always reading and learning about latest advances in psychiatry, and at a time when many were not attuned to the impact of cognitive deficits on functioning. Connie had read there was a treatment, and she wanted it for her college-educated child who she knew was not functioning at potential. In fact, cognitive impairment is very common in people with psychiatric illnesses. 95% of people with major depression, even after they've been taking antidepressants for a while, they have lingering cognitive dysfunction, even when their symptoms of depression improve. About 80% of people with schizophrenia have a cognitive dysfunction that places them below 91% of the general population. This has a negative impact on their ability to function in roles important to them, whether that is socializing, at work, at school, and managing their independent living. Keith Nuchterlein did a study where he tried to understand the major factors that impede people with recent onset schizophrenia from getting back to school and work. And as you can see here, he found that 52% of the variance in whether people return to work is accounted for by cognitive factors, things like attention and processing speed, working memory, and verbal memory. Let's take a moment to look at a video about someone who's struggling with cognitive impairment in the workplace.
So Matthew, last time we met, we talked a bit about your new job. You're working at a cafe, right?
Yeah, I like it for the most part. It can be pretty good when I'm doing stuff on my own, like stocking utensils and cleaning up and stocking napkins and stuff, but when I'm taking food orders, it can be pretty overwhelming.
Oh, what is happening when you get overwhelmed?
Hello, sir. Welcome to the lunch spot. May I take your order?
Yeah, let me get the turkey sandwich special. Tomatoes, no lettuce, pickles, whole wheat bread, light on the mayo, and a Coke.
Turkey sandwich special on whole wheat. And did you want anything else on that?
Yeah, tomatoes, pickles, and a little bit of mayo, and a Coke.
Tomatoes.
Tomatoes, pickles, mayo, and that's it.
Sorry, sir. Just punching this in.
Processing speed affects one's ability to take in information efficiently. Notice how Matthew struggles to work quickly, but also accurately.
Tomatoes, pickles, mayo. And did you want lettuce?
No, tomatoes, pickles, mayo, Coke.
Okay. Got it.
A multimedia show here. So people with schizophrenia and autism spectrum disorders also have deficits in social cognition. Social cognition broadly refers to the mental operations that underlie your social interactions. That can include perceiving people's facial affect, generating responses to behaviors and emotions, and understanding the intentions of others. These may manifest in difficulty taking another person's perspective, which is called theory of mind. A recent review of 112 studies involving over 7,000 people with schizophrenia and controls reported significant between group differences in social cognition, with a large effect size for emotion processing, social perception, and mentalizing. Social cognition impacts people in their everyday life, obviously in their social relationships, but also other aspects of functioning, like maintaining employment, because successful social interactions are so essential in the workplace. Let's take a look again at Matthew, who struggles with social cognitive deficits.
Let's find a place to eat. I'm hungry.
Sure, I can eat something.
Well, I've read about Joe's place. They have good hamburgers and great steaks.
I like salads and fish.
So shall we go to Joe's place? It's right around the corner.
Sure, okay. Maybe they have a salad.
So how does one go about treating these neuro and social cognitive deficits? That's something I've put a little bit of thought into. People very often write me and they ask, please tell me what computer program you use to treat cognition. And I write back, it's not computer programs that improve cognition. It is a therapeutic process, and that requires much more than any one computer program. You see, when you're struggling cognitively, it impacts so much else, not just your functioning, but also your self-esteem and motivation to do things, even the things you value. Treatment needs to take all of this into account. Most people with severe mental illnesses have complex needs. Seeing a patient might be the best way to demonstrate this. Let's meet a new person, Chris, to see why a comprehensive approach is needed.
Chris. Chris. Chris,
Yeah.
don't forget about those job applications. You said you were going to do that today.
I know. I'll do when I finish this round.
Chris, what are you doing? Those job applications aren't going to fill themselves out, you know?
I know.
Is that remote glued to your hand? Chris, you've got to stop being so lazy.
And you gotta stop nagging me all the time. There's no way I'm getting this done if she keeps bugging me.
Hey, do you have a second? Yeah, come on in. Thanks. My patient, who's really stuck, is coming in today. I wish I knew what to do with him. He just never gets anything done. He talks all the time about needing to get moving. If I bring up his goal to get a job, he just shuts down.
What's your sense of what's holding him back?
He was diagnosed with major depressive disorder 2 years ago, and he started antidepressants and therapy, and now his mood is better. But he has a really hard time getting activated, and he is very hard on himself.
Okay. Any issues with attention?
Yes, he was diagnosed with ADHD as a child. He abused stimulants, so he can't take them anymore. And he is really having a hard time focusing in group. He is fidgety. He can't track when other people are sharing.
He sounds like he could be a good fit for the executive functioning group. Why don't you suggest he attend the orientation so he can hear what we do and see for himself if it's something he wants to try?
Chris, like many patients, has very complex needs. If we just gave him cognitive remediation to help his attention, it would not address the large discrepancy between what he talks about doing and what he actually does. So what kind of therapy does he need? Let's go back to Connie and Steve. When we spoke, I explained that improving cognition with the goal of improving functioning involved much more than providing cognitive remediation. And I was not able to do that at Einstein, which is where I was when we spoke. Connie, not one to be deterred from a goal and always able to cut to the heart of a matter, figured out immediately how to resolve this seemingly intractable problem. Together with Jeff and Steve, she conceived the idea of the Lieber Recovery Clinic. And in 2007, I came to Columbia to make it a place where people used to accessing the best of psychiatric care could also access the best of recovery-oriented care. But what exactly is recovery-oriented care? Excellent recovery-oriented care is treatment with a focus that extends beyond symptoms to functioning, to quality of life and well-being. At the Lieber Recovery Clinic, we work not only with the identified patient, but with their social system, their work, and their school supports. The person is considered in the context of their environment, and we seek to address all the factors that will promote optimal functioning in their environment. It also means that there's a constant channeling of information from the bench to the clinic, breakthrough in our understanding of the basic biology of disease, coupled with research on how people use treatments, inform the way we deliver mental health recovery services in the here and now. It is essential to understand how people actually use their treatments. After all, Medications, medical devices, behavioral treatment strategies, no matter how efficacious, will be of little value if they're not taken. And that brings us to the topic of motivation. We spend quite a lot of time at the Lieber Recovery Center addressing motivation, and that's because it's such a common problem. The illness itself, the person's reactions to having an illness, and the environment can have a large impact on motivation. And the once eager, interested person may become withdrawn and disengaged. Let's hear from Chris about his struggles with motivation.
Well, for me, it's not that I don't know what to do. I just can't get myself to do it. I know I need to look for a job, but I usually just end up playing video games instead of looking. I mean, who's gonna want to hire me anyway? I have this huge gap in my resume, and I was 6. And they're probably looking for people who didn't make a mistake and have to leave. So.
Yeah, it sounds like you've had a lot of setbacks and you're not sure if this is going to work for you. And you're right to be skeptical and concerned because that actually shows that you care about this going well. It sounds like your confidence that things could turn out well, though, is pretty low. Am I right?
Yeah, I don't know if this is gonna happen. My mom's nagging on me all the time, and that kind of annoys me. It's like she wants this more than I do, so kind of makes me just want to say, forget it.
So the situation with your mom is having a pretty big impact on your motivation.
Yeah, I know.
Well, this group...
The motivational difficulties that Chris talked about are extremely common for individuals recovering from psychiatric illness. Part of what we address in the groups at the Lieber Recovery Clinic, in addition to practical skills building, are these motivational problems. Knowing skills intellectually is all well and good, but if you do not have the motivation to implement them, they're useless. One of the unique approaches our groups take to problems of motivation is not just to provide psychoeducation about the phenomenology of illnesses. We actually teach patients about motivational theory. We teach patients what influences them, whether they'll be motivated to do something. We teach them that they're more likely to complete a paper or work, task, or to socialize if there are an expectation of success, if they value the task as interesting or useful, and if in doing the task their psychological needs to feel competent, autonomous, and related to others will be met. And here you can see something we call the motivometer, where somebody is actually articulating why they would be motivated to do something. Sharing this information with patients empowers them and, in essence, helps them to become better self-managers. We also help clinicians and families identify concrete steps to support patients to maintain motivation. Importantly, we try to create a motivating therapeutic environment where patients experience success at the tasks they do, and where they have choice to identify tasks that are interesting and useful to them, and where they will be surrounded by people they find supportive. Connie took a deep interest not only in her own child's progress, but the progress of others at the clinic. She delighted in the stories of success and was always willing to talk as a peer parent to families who were struggling to help their child. Selfless, even in the face of illness, she was always by the phone, willing to drop everything to try and help just one more person. Together, Connie and I started the Connie Lieber Science to Practice Award to encourage dissemination of cognitive remediation therapies. And here you can see someone getting the award. Columbia Psychiatry has been at the forefront of cognitive remediation dissemination efforts. With the annual conference, Cognitive Remediation in Psychiatry, which is coming up this June 9th, and large-scale dissemination collaborations in Europe, Asia, Southeast Asia, Africa, Australia, and North America. The Lieber Recovery Clinic has been a model program for care systems that want to integrate cognitive remediation into their recovery-oriented treatment. Together, staff and patients host visitors from around the world who seek to learn how to offer personalized recovery treatments. In this way, we're able to carry on Connie's mission to touch the lives of many people actively struggling with psychiatric illness and to help them lead a productive, rewarding life. Thanks.
Good morning, and I have to say it's coming back here, having been here for many years, is always a heartwarming experience, and I'm pleased to see so many people from the department, friends, here with us today. And also, I'm delighted to participate after hearing 3 excellent presentations. I want to greet Steve, Sam, married, Janice. We also have Jeff Bornstein here, who's the president of the Brain and Behavior Research Foundation. And it was very nice to hear from Eric and Jonathan and Alice. So all in all, a lovely context for me to make a few comments. I was in Washington in 1984 and decided that the Reagan administration, which seems almost like a leftist organization compared to today, decided, to my disappointment, that in contrast to a promise they made to me, that they would not be making major boosts in brain research. And I decided to leave. I would look to come to what I thought would or could be the best psychiatry department in the country. And my interest focused on Columbia because I thought it fit the bill beautifully. In the mid-'80s, as professor and chair of psychiatry here at Columbia, I launched a series of symposia in which I encouraged the speakers to present psychiatric research in English. You hear me? In English. By which I mean, and some of you may have noticed, that scientists have a little bit of a tendency to get somewhat technological and somewhat detailed about things the audience has absolutely no notion what they're talking about. And we did that. So we decided to do it on a Saturday. It was a cold, wet Saturday morning, and I got concerned as to what kind of attendance we would have. The Columbia University auditorium for the meeting became packed with something close to 700 people. And 2 very friendly and attractive people approached me at the end of the conference and said they wanted to do something about psychiatric disorders. They wanted to support mental health programs, and particularly research. My instinct was that they were looking for an opportunity to do this in a very large scale, on a national, international basis. In any event, that was my introduction to Stephen and Connie Lieber. It led over years to Steve and Connie being leaders at one time or another of the organization NARSAD, which was begun in the mid-'80s in Washington, and went on to become the Brain and Behavior Research Foundation. In talking to Steve and Connie, it didn't take long to realize the extraordinary people to whom I was talking, their brilliance. Connie learned so much about mental illness and research, she probably would have been eligible to join the Society of Neuroscience. With the most poignant modesty, along with erudition, compassion, generosity, Connie answered questions, helped everyone, and as a team with Steve, made the Brain and Behavior Research Foundation the largest, and I would say the most effective psychiatric foundation, private foundation in the world for research, and the most collaborative, empathic, charitable institution I've ever seen. Our scientific grantees know that. They appreciate it. As a result, we can ask our scientists, we have 160 scientists on the review committees, to do almost anything. And for the Brain and Behavior Research Foundation, for the Liebers, they will do it. The Brain and Behavior Research Foundation, just to make a few comments. Number one, we went from an early dispute about whether we should spend our first $50,000 in the mid-'80s to a record in which we have now funded over $360 million in research. And given 5,000 or so grants, maybe a little bit, maybe even up higher than that, to people all over the world. We had some very experienced hands on the review committee, and Connie and Steve, the thing about them, they listen. And when they hear good advice, they take it. And so we had NIMH division heads, we had all kinds of people, chairs, Nobelists, researchers, and some of the principles that came out, I think, have been extremely important. One principle, people all over the world are eligible. The key thing is excellence in psychiatric research. Second, we had some very generous donors who decided to take care of the administrative costs of brain and behavior so that any additional dollar meant another dollar of actual psychiatric research. That doesn't happen in every philanthropy, as you might imagine. More recently, after a few unsuccessful attempts, we recruited Jeff Bornstein as president, and he's been an outstanding addition and leader in brain and behavior, and is busily expanding the reach and strength of the Brain and Behavior Research Foundation. As you've heard, the Liebers are and have always been extraordinary parents. They did everything they could with considerable success for Janice, their lovely daughter. They went on from the Brain and Behavior Research Foundation to create the Lieber Institute, whose board I happily chair at Johns Hopkins. And Steve is never short of ideas, I want you to know. And then established the Humanitarian Award in Mental Health, which is given to people who they are involved in mental health, but they're not involved in a very cubbyhole way. They are interested in broad aspects all related to mental health. So as an example, two of our recipients who received honorary awards was Mrs. Carter and Senator Kennedy. And other people who've received this included Dave Hamburg, marvelous psychiatrist, the head of the Institute of Medicine, and Vikram Patel from India. And again, that emphasizes the broad international scope of this, as well as the notion that we would have people who really merited the consideration. And that is ongoing now. I liked Alice's comments. She used two phrases which are right on target. Number one, selflessness. That really is a defining characteristic for Connie Lieber. And second, which I also enjoyed, was her saying that she was not easily deterred. Talking about Connie being easily deterred would be like saying that Trump would start functioning in a normal way. Connie's loss was devastating. Connie, and Alice said this as well, always focused on other people. We were taking care of her here, and very late in her illness, I went up to see her in her hospital room. Steve, as usual, was right there attending Connie. This extraordinary woman, beset by multiple illnesses, was in a very dire state. One might have expected some plaintive comments, but she looked at me, smiled, made no complaint, and said, "You know, Herb, the doctors and nurses are wonderful here. Would that more of our leaders emulated the model that she and Steve promulgate. Would that the people responsible for running all kinds of things in this country had the genuineness, thoughtfulness, and caring that Steve and Connie represent. Perhaps those leaders would have the kind of success the Liebers had in leading and supporting brain and behavior research to the point that it could claim the hundreds of millions of dollars it's given and chose the best people, young, middle, senior, to carry out the research. While Connie did this all with Steve, and she was responsive, as you've heard, and interactive to scientists, to people who were concerned about their families, wanted to know what to do about psychiatric illness, she also showed deep affection for children. We had a NARSAD conference a number of years ago, and my grandson, Alexander, of 3 years of age, was at the conference. And as we were getting ready, he started doing somersaults around the, and Connie just loved it. Connie won his way into her heart. You might be interested, and I was just thinking about it recently, Connie had a deep interest in architecture. And maybe she influenced him because my grandson is now a third-year student in architecture school. But it wasn't only Alexander who won their way into Connie's heart. Connie's heart is a very big heart. It is one that we will miss and hold in hearts forever. This is a remarkable woman married to a remarkable man, and these two have created an organization which is unique in its positiveness, its honesty, and its looking for excellence, and its desire to do something about psychiatric illness. And they're going to help us all succeed. Thank you.
Herb, thank you for those really poignant and profound comments on Connie and kind of the career that she and Steve had philanthropically and the legacy that she leaves. I think it frames things in a way that is very timely in that, to change a culture, to change society, to change the course of civilization is a huge undertaking, and one thinks that it's really something that only governments can do. And unfortunately, governments aren't always optimally functional or led. But it's amazing how individuals can impact these macro issues. We've seen it historically. Dorothea Dix for the mental illness Mary Lasker with cancer, various individuals, Jim Simons with autism, Michael Milken with prostate cancer. You know, the Liebers have done similarly that kind of thing with regard to mental illness. And it basically illustrates the fact that individuals where there's the commitment, the wherewithal, the enlightened vision can impact things in a way that's measurable and alters the lives of people for the better. So I think, you know, given the fact that we've expired the usual amount of time for Grand Round's presentations, that we need to forego the discussion and questions, except for the fact that we'll continue that at lunch upstairs for people that are invited to attend. And if you're not invited but really want to come, why don't you come and we'll see if we can fit everybody in, if there's food enough to go around. But I just want to conclude by thanking the speakers on, you know, the presentations, that each of which could have occupied the whole hour and a half. Thank for the Lieber family for all they've done and for being here today. And Jeff Bornstein, you know, you're carrying on your shoulders a lot of responsibility and expectations, but you've been doing great so far. So keep up the good work and know that we'll always be there to support you. And let me thank everybody for their attendance, and we'll adjourn and reconvene upstairs in the multipurpose room.